Okay, let's do a deep dive today into something really fundamental. How our blood ages.
Right. And not just, counting birthdays, but what's happening deep down cell by cell.
Exactly. And we're looking at a really interesting 2023 study from Nature highlighted by Lifespan.io. It introduces something new.
Yeah, a tool called Epi-Clone. And it's giving us a completely fresh perspective on blood aging.
Epi-Clone. So what is it? Is it like another genetic thing?
Not exactly genetic modification, no. It's a single-cell methylation tool. It basically reads the natural DNA methylation patterns already in your cells.
Methylation patterns. Like little flags on the DNA.
Kind of, yeah. Think of them as tiny built-in barcodes unique to cell lineages. Epi-Clone uses these barcodes to trace a cell's history where it came from, its ancestors, without needing to tag it artificially.
Ah, okay. So it's reading the cell's own history log.
Precisely. And doing it one cell at a time. That's the key.
Why is that single cell view so important here?
Because it lets us zoom in on the really crucial players. The hematopoietic stem cells, the HSCs, the ones that make all our blood.
Right, the master cells. And their decline is a big part of aging, isn't it?
A huge part. Epi-Clone lets us follow individual HSCs and see what happens to them and the cells they produce over time.
So what does happen as blood ages? Does it just slow down?
It's more about diversity, actually. You start with a really varied mix of blood cells coming from lots of different stem cell families. Healthy.
But that changes.
It can, yeah. Over time, the system can become less diverse, dominated by just a few large families, or clones. That's called oligoclonality.
Logonality. And sometimes that's driven by specific genetic mistakes, right? Mutations.
Exactly. You mentioned mutations like DNMT3A or TET2. These are surprisingly common as we get older.
And they affect methylation, don't they? The very thing Epi-Clone measures.
They do. These mutations can give certain stem cells a kind of competitive advantage, letting their clones expand and take over more space, crowding others out.
And when that happens, that expansion, that's clonal hematopoiesis, CH.
That's CH, yeah.
Which isn't just some abstract biological process. It's linked to real health problems.
Very much so. Increased risk of cardiovascular disease, certain blood cancers like leukemia. It's a significant marker of age-related risk.
But wait, are all these expanding clones automatically bad news?
That's a great point. No, not necessarily. Some clones might just be expanded without being particularly dangerous, at least not initially. Others might be acquiring more worrying changes.
Okay, so that's where Epi-Clone comes in again, to tell the difference.
Precisely. By reading those detailed methylation barcodes on single cells, it can help distinguish. Is it just a big clone, or is it a big clone showing signs of trouble? It gives us that finer resolution.
So stepping back, Epi-Clone is giving us this incredibly detailed picture of blood aging, tracking individual lineages.
Yeah, it's like moving from a blurry photo of the whole blood system to a high-definition movie, tracking each individual actor. We can see who's expanding, who's declining, and maybe why.
What could this mean, practically speaking, for, say, monitoring health as we age?
Well, think about current epigenetic clocks, like blood age or DNA methylation. They measure average methylation across millions of cells. Good. But they miss this clonal story.
Ah, so Epi-Clone could add that crucial detail, make those aging clocks much more informative.
Potentially, yes. It could add a whole new dimension, revealing not just overall biological age, but the specific clonal dynamics driving it. Much more precise monitoring.
And what about treatments? Does knowing about specific clones open up new possibilities?
Absolutely. That's maybe the most exciting part. Instead of broad treatments, you could aim for highly targeted interventions.
Like what?
Well, maybe using senolytics to clear out specific bad actor clones that are causing inflammation. Or for clones with specific mutations, like TET2.
Right, you mentioned TET2.
Yeah, there's research suggesting things like high-dose vitamin C might selectively disadvantage those TET2 mutated cells through metabolic effects.
Wow, that's specific.
Or using drugs like BCL2 inhibitors, which block survival signals, perhaps targeting clones that are starting to look pre-leukemic. Really precise stuff.
It sounds almost like science fiction targeting individual cell families based on their history.
It's getting closer. Of course, there are challenges. Single-cell tech is still, costly and complex. We need more validation. We need to understand how well blood reflects the bone marrow.
Sure, the usual hurdles with cutting-edge science.
Right. But the underlying promise, a much more proactive, personalized way to manage aging, starting with the blood system, it feels very real.
It's quite amazing, isn't it, that so much history and future potential is written in these subtle methylation marks on individual cells.
It really is. The complexity hidden within something we often take for granted.
So it leaves us with a pretty big question, I think. As we develop tools like Epi-Clone and get better at reading these deep cellular histories, how is that going to change how we approach aging altogether? What really stands out to you from this dive?